Hello everyone, this is Mr. Jackson, and today we're going to be going into Lesson 5. This is going to be the first of two parts for Lesson 5, and in this one we're going to be talking about DNA structure and its overall function. So you'll know that you've mastered this lesson at the end when you're able to identify the components or those individual building blocks of DNA. So what are those building blocks? You should be able to explain what those complementary base pairing rules mean. And that is a very crucial learning objective that we're going to be utilizing for the rest of the unit in genetics. So very important that we know what that rule is. And then you should be able to describe the role or roles of DNA in heredity. And we'll go ahead and get started. Now remember, whenever we talk about the four macromolecules, we had one of those types of macromolecules that were the nucleic acids. And this is the macromolecule that holds our genetic material. So DNA is one example of these nucleic acids. And it's important to understand that these nucleic acids contain genes. These are sections of DNA, so small snippets of DNA that basically serve as a blueprint or the specific instructions for making a specific protein, right? So every protein that we have in our body, all of which have functional roles, structural roles in the body, all of those are basically coded for by sections of your DNA called genes. And these are located at certain points along each of your chromosomes. So proteins are what ultimately will carry out all the cellular activity in your body. They're what give us our traits. You are a combination of your proteins, just as you are a combination of your traits. And we have two main types of nucleic acids. We will be using the short form, DNA, RNA, but it's important to understand, so what that actually stands for. DNA stands for deoxyribonucleic acid, and I've highlighted the letters within that term that basically give it its name, right? It is called deoxyribonucleic acid because it has a sugar in its in this nucleic acid that is called deoxyribose. You're not going to have to remember that specifically, but that is how we get the name. RNA, it's a different sugar. It's a ribose sugar. And so instead of deoxyribose, now we're going to call RNA ribonucleic acid. Okay, but DNA and RNA are going to be the ones we focus on in this unit. DNA is the one we're focusing on today. So the monomer, or the individual building blocks, think back to that first learning objective. We want to make sure that we understand that these individual building blocks of nucleic acids, we call these nucleotides. Okay, so nucleotides are the building blocks of the nucleic acids, the DNA and the RNA. And over to the right, as I'm going through this, you can actually see examples of DNAs for different nucleotides. We also have a fifth one that applies to RNA, but we'll get to that in a future lesson. So these nucleotides, they're made up of three parts. In purple, you see that we have a sugar. Again, for DNA, that's going to be called deoxyribose. For RNA, it's called ribose. You do not have to remember that specifically. Just remember that we have a sugar for each. We have a phosphate group here in pink. And we have one of four nitrogenous bases. Now, the official names of these four bases are adenine, guanine, cytosine, and thymine. But for our purposes moving forward, it is nice to know those, but I'll always give you the ability to just simply go off of the letter for those nitrogenous bases. So instead of adenine, just remember the letter A. Right, so we've got A, G, C, and T. Whenever we get to RNA, we'll also include uracil. That will replace our thymine. So instead of a T, we'll have a U. We'll have plenty of time to explore what that means. So overall DNA structure, okay? Now, DNA structure is, it looks like sort of like a twisted ladder. If you look over here all the way to the right, this is exactly what a double-stranded DNA molecule might look like if it were unwound and stretched out. Okay, so we call this official structure a double helix. It's two strands that create a helical shape, and it looks like if I took a ladder and twisted it in a spiral. And that's actually going to help a lot, because by doing so, we're actually able to condense that down whenever we don't actively need to access each of those bases where you see those letters on the inside. Okay, so it's important to understand that these sugars and these phosphates essentially create its backbone, or its overall structure, like its linear structure. So these are going to bond together in order to hold the overall DNA molecule together. And then in the middle, we have these nitrogenous bases that bond with what would be relatively weak hydrogen bonds. The relatively weak aspect of that we will definitely go back to and review, because it actually is for a very specific purpose why these bonds in the middle are so weak. But all other bonds, like for example, the nitrogenous base bonding with the sugar, and the phosphate bonding with the sugar, and this phosphate bonding with the next sugar for the next nucleotide, all of those other bonds that are not in the middle are way stronger. And those are covalent bonds, getting a little bit back into that basic biochemistry. But really, what we need to understand is, structurally, if something is going to become separated, it's going to be between these two bases in the middle. And that is going to be crucial to understand as we go through the process of DNA replication. Very important rule. So this is going into that complementary base pairing rule. So it's important to understand that whenever we see two strands bonded in the middle, nitrogenous base to nitrogenous base, we are always going to see that our adenines, or A's, always bond with thymines, or T's. A's are always with T's. Okay, so if you've got an A on the left, you will always have a T on the right, and vice versa. Easy way to remember this, you might have seen in some other videos that we've reviewed. Easy way to remember that is the apple is in the tree. Okay, so the apple is in the tree, A's with T's. And then you also always have guanines bonding only with cytosines. So G's and C's. And the easy way to remember that is car in the garage. So C with G, car in the garage. So apples in the tree, car in the garage. And the official term for this, based on the scientist who established that these base pairs are always together, they're always in the same amounts, that was Chargaff. And so we could call it Chargaff's rule, but we're going to just call it the complementary base pairing rule. You can think of the A's and T's complementing each other, or the G's and C's complementing each other, because they always pair together. Really good way to remember the DNA structure is by comparing it with the RNA structure. Now, we're going to use both extensively in this unit. So, whereas we just said that the overall structure of DNA is a double-stranded helical structure, so a double helix, like a twisted ladder, RNA, ribonucleic acid, is just a single strand of these nucleotides, and all of the bases that would typically in the middle, those are going to be outwardly exposed. You can see I can easily access any of these bases where they would normally be bonded, because they no longer are. They're accessible, they're exposed. RNA bases are able to bind with existing DNA bases, right? So that means that we're still going to apply these base pairing rules. The only difference is that RNA doesn't use the nitrogenous base thymine. There are no T's when it comes to RNA. And instead of RNA, we still want to have something that can always pair with our A. So we had A with T, apple in the tree. Now we're going to be replacing thymine with uracil. So this T will always become a U whenever it's with RNA. And it's still an easy way to remember it. Don't lose the apple, right? It's just kind of like now the apple fell and it's under the tree, right? So for RNA, it's apple under the tree. And then with this structure, we're also still going to have the same base pairing rule. Guanine and cytosine still go together. So importantly, based on this base pairing rule, if I'm going along this RNA strand, I would know that because I have a uracil here, a U, that would bond nicely with an A on the other side, a C with a G, an A with a U, an A with a U, and so on and so forth, building out using that base pairing rule. Gave you a little table here for you to make those quick comparisons between DNA and RNA. Again, really important things to remember here are the types of bases that we use. All are going to be the same except for the T and the U. T with DNA, U with RNA. The type of sugar is what ultimately gives it its name and makes it different. So we have a deoxyribose sugar giving it DNA, ribose as the sugar for RNA. And then the shape, we have a double helix. You can see that up top, kind of like a twisted ladder. And then a single strand of RNA, and those bases are all exposed. And finally, we want to make sure that we are perfectly clear on what this structure allows in terms of DNA's overall function. And so remember that DNA can do three big things. The first thing is DNA stores all of our genetic information, right? So we said that DNA is basically this long double strand of all of our nucleotide bases. And if you take enough of those letters together, you can put them together into what we call genes, kind of like words in an overall sentence. And these genes all mean a specific thing. They code for a specific protein. That is how our DNA in every single cell of your body is going to actually hold and store all of the genetic info, all of the instructions for every cell to make any protein to give you any trait that you have. Also, another function is that it can copy itself, right? So we're going to get to in part 2 this enormous process, this ultimately important process called DNA replication, where we start making those copies so that all of the cells in our body can have all of the information they need. And so we know that every time a cell divides, it has to make an exact copy of all of its genetic information. And then finally, there's this ability for DNA to express itself, right? This is another way of saying that, okay, we have all this info, let's put it to work, right? So we're going to use the instructions for DNA to produce proteins and to produce those functional units that will ultimately keep our bodies running smoothly. Okay, so again, storing info, copying info, and then expressing that info is key to the role of DNA. And so that's going to conclude for part 1, where we went over DNA structure and function. And I hope you found this helpful, and we'll see you in part 2.